Why do individual organisms die, but their genes live on? This is the heart of the immortality by reproduction paradox, a puzzling idea suggesting that organisms might be designed to die so their genes can live forever. Evolution appears to favor genetic survival over individual longevity, meaning your body is just a temporary vehicle for passing along your DNA. From this perspective, death is not a flaw but a feature. It clears the way for new generations, allowing genes to adapt and thrive in changing environments. Example, bacteria reproduce by division and can theoretically live forever. Yet complex organisms with sexual reproduction are built with aging and programmed death. Why? Because it's not the individual that matters most, it's the gene. But how does reproduction lead to genetic immortality? When organisms reproduce, their genetic code is copied and passed on, often with small mutations that fuel evolution. This continuous replication allows genes to persist far beyond the lifespan of any one organism. In this sense, your DNA is functionally immortal, even as your body ages and dies. Analogy, it is like a relay race where the baton, your genes, keeps moving forward, even though each runner eventually stops. The paradox challenges our view of life and death by suggesting that mortality is not a failure of biology, but a strategy for eternal genetic survival. Are individual organisms programmed to die so that their genes may persist indefinitely through offspring? From a gene-centric view of evolution, the ultimate goal of life is not the survival of any one body, but the propagation of its genetic information. Yet individual organisms invest vast resources in maintenance and repair, seemingly striving against decay, only to face an inevitable demise. This tension between somatic endurance and reproductive renewal gives rise to the immortality by reproduction paradox. How can the drive for individual survival coexist with a life cycle that mandates death for the sake of gene continuity? So far, so good, until we examine the evolutionary logic that underpins senescence and programmed death. According to the disposable soma theory, organisms allocate finite energy between reproduction and bodily upkeep. Once reproductive fitness peaks, natural selection biases energy investment toward offspring production rather than lifelong tissue maintenance. In this framework, aging and mortality are not malfunctions, but evolved trade-offs. Genes promoting early life fecundity can afford to tolerate or even favor later life deterioration because once the genetic legacy is secured, further somatic survival offers diminishing returns in evolutionary terms. At even smaller scales, molecular mechanisms vividly illustrate this programmed sacrifice. Telomere shortening, accumulation of DNA damage, and regulated apoptosis exemplify built-in countdowns to cellular and ultimately organismal death. While repair pathways work tirelessly, they cannot fully forestall the entropy of replication errors and oxidative stress. In some species, like Semelparous salmon or annual plants, death follows a single, massive reproductive event, driven by hormonal cascades that actively dismantle soma in favor of gamete investment. These molecular death clocks ensure that resources flow from parent to progeny with maximal efficiency, even if it means self-destruction. And then there is the matter of ecological and population dynamics. In crowded or resource-limited environments, the timely removal of older individuals prevents competition with the young, enhancing overall population fitness. Programmed death can synchronize generations, reduce disease transmission, and allow rapid adaptation to changing conditions. For social species, elder mortality may even shape cooperative behaviors, as post-reproductive individuals contribute to kin success through grandmother effects rather than direct reproduction. Through these community-level benefits, the paradox resolves. Individual mortality becomes an adaptive feature that secures the long-term immortality of the gene pool. Ultimately, the immortality by reproduction paradox illuminates a profound principle of life. While bodies are transient, the genetic information they carry can achieve a form of perpetuity. Death is not merely an unavoidable breakdown, but a carefully orchestrated stage in the life cycle, one that balances the molecular costs of maintenance against the evolutionary imperative of gene propagation. In this light, individual mortality and gene immortality emerge not as contradictions, but as two sides of nature's strategy for enduring success. The immortality by reproduction paradox asserts that individual organisms are intrinsically mortal, yet their genes can persist indefinitely through successive generations. Natural selection appears to favor death at the organismal level because it facilitates resource turnover, reduces competition with offspring, and accelerates adaptation. In this view, mortality is not a design flaw, but an evolved feature ensuring the long-term survival and diversification of genetic lineages. The paradox challenges our instinctive yearning for eternal life by revealing that individual death may be the price genes pay for enduring immortality. Pacific salmon die immediately after spawning, yet their genetic material lives on in countless fry downstream. It is like a baton in a relay race. Each runner must drop the baton at the finish line so the next runner can continue the race. Classical biology treats death as the inevitable endpoint of an organism's life cycle, but genes themselves are replicated anew each generation. Richard Dawkins' selfish gene concept reframes organisms as temporary vehicles built to ferry genes into the future. From this perspective, organisms are disposable survival machines, while the true units of selection, the genes, achieve a form of immortality through reproduction. This distinction reveals how the lifespan of the individual can be decoupled from the persistence of genetic information. Germ cells in mammals maintain DNA integrity across generations, even though somatic cells age and die. It is like software running on hardware. The hardware may crash or be replaced, but the software endures through installations on new machines. At the molecular level, organisms invest resources in reproduction at the cost of somatic maintenance, creating a trade-off between lifespan and fecundity. Repair mechanisms and stress responses are energetically expensive, so allocating energy to egg or sperm production often shortens individual survival. This allocation strategy maximizes gene propagation, even when it accelerates. The paradox arises because what benefits the gene pool can harm the individual organism. Fruit flies engineered for increased stress resistance live longer, but produce fewer offspring compared to wild-type flies. It is like a business choosing to invest all its profits into expansion rather than maintenance. Growth continues, but the original infrastructure suffers. In essence, the progressive decline in physiological function is driven by genetic pathways such as telomere shortening, DNA damage accumulation, and apoptosis. In some species, programmed death, e.g. simulparity, is an adaptive mechanism where organisms die immediately after reproduction to free resources for their progeny. These mechanisms underscore that death can be an evolved feature rather than an accidental failure. The paradox lies in recognizing that genetic programs actively orchestrate organismal demise to optimize lineage success. Pacific octopuses undergo rapid degeneration and death after brooding their eggs, a behavior regulated by hormonal cascades. It is like a disposable launch vehicle that self-destructs after delivering its payload to orbit, ensuring no interference with subsequent missions. Inclusive fitness theory shows that genes can increase their representation both through direct reproduction and by aiding relatives who share those genes. Mortality of older generations can enhance the survival prospects of kin by freeing up territory, nutrients, or parental care. Thus, programmed aging and death can be favored if they boost the overall genetic contribution to future generations. The paradox highlights how individual sacrifice can serve the broader genetic network. Elder wolves often leave territories to their offspring by dying or dispersing, thereby improving pack success. It is like a senior employee taking early retirement to provide career advancement opportunities for junior colleagues, ultimately strengthening the company's talent pool. Life history theory demonstrates that small changes in mortality risk or reproductive timing can produce disproportionate shifts in population growth and genetic spread. Species exhibit a continuum from live fast, die young to slow and steady strategies, depending on ecological pressures. These nonlinear responses reveal how mortality schedules are finely tuned by evolution to maximize gene transmission under varying conditions. The paradox emerges from the observation that minor tweaks to lifespan can have major evolutionary payoffs. Annual plants complete their entire life cycle in months, while perennials live decades, each strategy optimized for different environments. It is like adjusting the throttle on an engine. Slightly increasing RPMs can dramatically boost power output, but at the cost of engine wear. Understanding this paradox informs conservation strategies by recognizing the ecological role of senescent individuals in nutrient cycling and habitat structure. The death of mature organisms often releases resources that underpin ecosystem productivity and support juvenile cohorts. Ignoring these dynamics can lead to mismanagement of populations and habitats. The paradox thus bridges individual-level physiology with broader ecosystem function. The carcasses of spawning salmon deliver marine diverg nutrients to freshwater ecosystems, boosting plant and insect populations. It is like retiring old fruit trees in an orchard to enrich the soil for the next planting cycle. Accurately capturing the interplay between individual death and gene persistence demands models that integrate genetics, physiology, and ecology across scales. Simplistic models risk overlooking key feedbacks such as kin selection, resource competition, and demographic stochasticity. Parameterizing rates of senescence and reproduction in diverse environments adds further complexity. These challenges underscore why predicting evolutionarily stable lifespans remains an open problem. Models of nematode aging must reconcile laboratory-observed longevity mutants with field-measured survival under fluctuating resources. It is like forecasting traffic flow by modeling individual driver behaviors, road quality, and weather, all at once. Emerging research on programmed cell death, epigenetic inheritance, and transgenerational effects promises to refine our understanding of this paradox. Advances in single-cell sequencing and long-term ecological monitoring will reveal how mortality strategies evolve in real time. These insights could inform biomedical approaches to aging and fertility, as well as ecosystem management. Ultimately, unraveling the immortality by reproduction paradox may reshape our views on life, death, and the continuity of genetic lineages. Studies on tardigrades, repeatable dormancy, and cryptobiosis explore alternate routes to gene persistence beyond conventional reproduction. It is like decoding the firmware of a machine that can reboot itself in different modes to ensure its core programming endures under any condition.